Hi everyone, my name is Hang Li. On behalf of PPRI team, I'm delighted to be here to talk about the activities and results that we've done in the trip over the past 2 years. For Objective 3, we have 6 main contents, and so far we have been implementing 3 of them. I have here 2 tables showing achievements in summary that reported in the annual reports of each year. Basically, achievements are related to training, surveillance, and diagnostic. I'm gonna go first with surveillance of disease. The 2 target diseases of this project are cassava mosaic disease and cassava witch's broom disease. During the 2 consecutive years, PPRI team has conducted surveys and sampling on 479 fields. All data is shared on a common platform. For data analysis, 39,890 observations were shown on patch display that conducted by 3 country teams, in which there are 24,345 observations in Vietnam alone. For surveillance of cassava mosaic disease, this is the Incidence Maps of CMD, showing data collected in 2014 and the 2 consecutive years of 2020 and 2021. We can see that CMD hasn't appeared yet in Vietnam in 2014, and in fact, CMD is just first reported in Vietnam since 2016, and from then on, it has become one of the most serious threats to cassava production. I have color-coded shown CMD hotspots across Vietnam. CMD is spreading step by step from south to north. For surveillance of cassava witch's broom disease, it is also shown through incidence maps. CWBD is first recorded since 2008 in Vietnam, and then quick spread and become a serious disease from 2010 to 2015. However, it was almost disappeared from 2015 to 2020. Recently, field surveillance was carried out in late April 2021. We have got notable results that we found down CWBD on the fields, and it was really coming back after long absence. I'm going to move to some activities in laboratory. Diagnostic. In 2020, we did test some samples with CMD by PCR. Because of the large samples, we had to use the pooling sample method, but actually the results were not very well at all. From my experience, the bad results of PCR were caused by DNA quality after extraction. That is why in 2021, we focused on finding a method for rapid, inexpensive, and efficient isolation of high-quality DNA from cassava leaves, suitable for PCR pooled sample test. And now we have a complete protocol for this. This method is based on CTAB method, and we call it modified CTAB method. In PPRI lab, for detection of CMD, we use PCR with primers specific for Sri Lanka cassava mosaic disease, and for detection of CWBD, we use nested PCR. And also, we recently do an experiment to detect viral diseases from cassava cutting stems. It is a little bit more difficult than detection of viral disease from leaf, but detection of disease on cutting stems is very, very important in the selection of disease-free varieties for new crop. And we also have some interesting results for these experiments. I'm gonna go with some results related to insect vectors of CMD. For the surveillance of whitefly, sampling of whitefly was also designed during surveys of disease, followed by the protocol of CAD. The photo data that taken on the second youngest leaf were shared on a common platform and also shown on patch displays. During the surveys in 2020, we observed very, very little whitefly on the field. In the surveys in 2021, Let's see the correlation between the Whitefly ratio and CMD. The ratio of CMD is kind of proportional to the rate of Whitefly. According to the protocol of CEAT, we take a photo of the second youngest leaf based on the prevalence of whitefly in Latin America and Uganda. But in 2021 surveys, we did an experiment of evaluating the correlation between the cassava leaves and whiteflies in Vietnam by determining the density of whiteflies on the first 5 youngest leaves on the 30 plants from different fields. And the very interesting result that the prevalence of whitefly in Vietnam is the 4th leaf. In 2021, we also carry out the diversity of whitefly in Vietnam. Sequencing of 1886 whitefly samples collated during 2020 and 2021 surveys for phylogenetic analysis based on anticoid gene. And this is the short protocol diagram for preparation of whitefly for sequencing applied in PPRI lab. We have got 67 valuable sequences divided into 3 regional groups, including samples from the south, samples from the north, and samples from the center. Here we have the phylogenetic analysis of whitefly samples collated from the northern provinces, along with the sequences of previously identified species or biotypes obtained from database. And we only detected Asia 2.1 and Asia 2.6 biotypes. Samples collated from the central provinces, we also only detected Asia 2.1 and Asia 2.6 biotypes. The last one, for whiteflies collated from the southern provinces, we only detected Asia 2.1 biotypes. So the whitefly biotype in Vietnam is mainly Asia 2.1, and meaning to say, Asia 2.1 is an efficient vector of SLCMV in Vietnam. And above are the notable results that PPRI team has achieved in the first 2 years of the project. Thank you so much for listening, and I'm willing to answer any question from you. Thank you.